and welcome to a Nutrition Insights interview series. My name is Vena Patel. I'm a senior journalist, and today I have the pleasure of speaking with an expert from the Boyce Thompson Institute and Cornell University to explore the world of gut bacteria and its impact on health. So thank you so much for joining us. And to start, could you please introduce yourself and tell us a little bit about your work?
Yeah, so I'm a chemist and started identifying new molecules from nature during my PhD 20 years ago or so, and then became interested in the interactions of the human gut microbiome with the host, with the rest of the body.
Mhm.
And what research in the past 15 or 20 years or so has revealed is that these interactions really rely on small molecules to a large extent, small metabolites, small compounds that, for the most part, are new molecules that are new to science and have not previously been characterized. And I felt this is a huge opportunity for discovery, to learn something fascinating about biology, about how biology uses chemistry, and
Mhm.
also an opportunity to perhaps develop new types of compounds, new types of approaches to treat human disease, because it has become very clear that these gut microbes play a central role for many human diseases.
Mhm. Well, thank you so much for your introduction and for being here. Let's dive right into the questions. My first question for you is about the gut microbiome, which is a really hot topic right now in nutrition science. So, what are some of the biggest misconceptions that you have seen, and how is it discussed in mainstream health advice?
I think the risk always is oversimplification. I think when I grew up, the microbes were considered to be something dangerous, right? You had a bacterial infection, and that was, of course, potentially a very serious matter. Now, there's this idea that there are specific microbes that are good and bad for human health. And that is certainly true. There are specific bacteria that can affect human health negatively. That's very clear. They can cause serious infections. There are specific bacteria that also may be beneficial. But what is lost in this view is that it's really about the interaction of the bacteria with the host. And that even more so, it's further complicated by the different bacteria interacting with each other and the host, right? So it's many different players that interact together. And what the outcome is for human health really depends on uh, the entire spectrum of microbes that we have in the body, and also the specific genetic makeup of the host, right? So we all are different. We have different gene expression patterns, and that matters for which microbes will have beneficial or perhaps detrimental effects. Important question is diet, right? So
Diet. Yeah.
the diet, yeah. So if we talk about microbes, what they really do in the gut
Yeah.
depends on the diet.
Yeah.
So we have a system that is further complicated by decisions that we make about what we eat, right? So we have different nutrient and different substances getting
Hmm.
into this system, and then microbes may respond in completely different ways, depending on what that is, how it is digested, what compounds, chemicals are available in the foods that we ingest. And that again affects the phenotypes that are associated with it. So you may have one microbe in one human where it's completely innocuous and doesn't do anything bad. And then you have another human that has a different diet or a different genetic makeup. And then you get very, very serious changes. And these can affect the immune system. And we have many cases where there's allergic responses, there's inflammatory responses in the gut that can lead to very serious disease, inflammatory bowel disease, colitis, or metabolic disease, where people just gain fat, where the liver accumulates fat, and we get very, very serious conditions as a result.
Yeah. And so you mentioned how every individual will have a different microbiome with different bacterial strains. So do you think it makes sense to talk about dietary trends and whether certain dietary trends are overhyped, or or dietary trends that need more attention? Does it make sense to think about these trends if our microbiomes are so different?
I think it's dangerous to follow any trend, if you will, right? So I think maybe 20, 30 years ago, people were told not to eat any fat because it would make them fat. And today, I think we know that maybe too much sugar is not a good idea. So we go through these phases, right, where people are told to do one thing or another. And the truth is that it really depends on many factors what's good for an individual.
Mm-hmm.
So I'd be cautious to follow any trend. And rather, I think looking forward, what we're probably headed toward is a more personalized approach to medicine, where we consider the specific genetic makeup of the individual, then the existing microbiome, and perhaps from that one can develop dietary recommendations, considering the specific health parameters that this individual is exhibiting. So I think it's best to stay away from trends and rather see what's good for yourself, right? So I think that's maybe a recommendation that certainly makes sense to follow.
Yeah, that makes sense. So let's talk a bit more about your recent research, which suggests that the gut bacteria can help, a certain gut bacteria can help regulate cholesterol and fat metabolism. So how could this discovery change the way that we approach conditions like fatty liver disease?
Yeah, so what we found is that and I think I mentioned already earlier that we have to move away from this idea that the microbes do something specific and then the host is just a passive bystander. So our research really shows that the microbes and the host, by the host I mean the human, right? It's the human, or in our case, in our research case, the mouse. Um, that they interact. There's biochemistry going on in the bacteria and in the host. And it's not that these two biochemical worlds don't interact, they're intricately connected. And
Mm-hmm.
to some extent, that was known. It was known that the host makes certain acids that get via the bile duct put into the small intestine, where then the microbiota convert them. They convert them into what we call free bile acids. And these free bile acids, they had been known for quite some time to be agonists of nuclear receptors that govern many aspects of metabolism and immune responses. And so in this model, the microbes would produce free bile acids that then would shift the host metabolism and its immune system in one specific direction, right? You have these
Mm-hmm.
free bile acids, they bind to the nuclear receptor, and then the host physiology shifts in one specific direction.
Mm-hmm.
So this model basically puts the microbes in charge, right? They produce the free bile acids and the host just responds and the physiology changes. And that can include reduced bile acid production. And the important point here is that the bile acids are made from cholesterol. In
Okay.
fact, one of the major avenues by which the body gets rid of excess cholesterol is by converting it into bile acid.
Mm-hmm.
So the way the traditional model works is that the microbe produce free bile acids that feed back on the host and actually reduce conversion of cholesterol into bile acids. So here the microbes have a role that reduces the biochemical elimination of cholesterol. And
Hmm.
this has important consequences for liver fat content. It usually is increased, right? If you have high cholesterol in most models, there's many details to that. Cholesterol itself is something we could talk about for 5 hours, probably. Um, uh, but certainly, overall cholesterol elimination is reduced because the microbiota do this. They produce these free bile acids from the precursors that the host. So that's the traditional one. Now, what we found is that the host converts these microbe-derived free bile acids into yet another class of bile acid derivatives, a new class of bile acid derivatives that had been overlooked, that people just hadn't seen because it's made in the small intestine. Normally, bile acids are made in the liver, and that's correct. They're made there. And what we've shown is that the microbe-derived free bile acids are converted into yet another class bile acids in the small intestine. And these new derivatives, we call them bile acid MCY conjugates. It's an abbreviation for a chemical moiety that's characteristic of these newly identified derivatives, that these host-produced bile acids have the opposite activity of the microbe. So the idea here is that the host converts what the microbes make into a new molecule that has the opposite meaning, has signaling properties that directly oppose the signaling properties of what the microbes. So basically, the host pushes back. And the effect of that is that now bile acid production in the liver is increased. That conversion of cholesterol into bile acids is increased. Yeah, so it's basically a host and microbe pushing into different directions. The
Yeah.
microbes produce free bile acids that reduces conversion of cholesterol into free bile acids, and the host makes derivatives that increases conversion cost, right? So it's two
Hmm.
different types of feedback here that antagonize each other.
Hmm. But in a way, they are working together in some sense, right? It's
They are, I would say, they're balancing each other, right? So
yeah.
of course everything is interdependent. So we showed that the host production of these new derivatives depends on how much the microbes have made of the free bile. So if the microbes make a lot of the free bile, which would push the system into one direction, then the biochemical answer of the host is